Hello, my name is Fernandez, and I will be covering the article, Vitamin D Regulates Microbiome-Dependent Cancer Immunity. First, let's start off with some background information. We'll first talk about globulin, then the vitamin D pathway and its importance, the gut microbiome, and some immunotherapy terms. Firstly, globulin. Globulin is a group of proteins that's found in the blood, and it has a role with secreted gelsolin, SGSN. It also has a secondary role with the involvement in the vitamin D pathway, which we will talk about on later slides. If you look here, let's start off with the CDC1 cells. CDC1 cells are conventional dendritic cells, which binds to the DNGR1, indicated right here. This allows for the binding of F-actin, which are things released by dead cancer cells. The SDC1 takes this side here and attaches it to the CD8 receptors on T cells. This allows for the activation of the immune response. The problem here is SGSN, indicated here, takes away the F-actin, preventing this whole thing from occurring. Now, you might be asking, how does globulin relate with this? Globulin allows for SGSN to find F-actin, increasing this. Now for the vitamin D pathway. There's two forms of vitamin D, vitamin D2 and vitamin D3. While both can be found from our diet, only vitamin D3 can be found with sunlight exposure. Both of them go into the liver, and from there, they're converted into 25-OHD, also known as calciferol. This is what we'll be focusing on, as this is what circulates through our bloodstream. Here is where GC kicks in, as GC has an N-terminus where this can attach, preventing circulation. Also, once it circulates, it should be noted that when passing through the kidneys, it gets converted into 125-OH2D, which might be mentioned later. Now for the vitamin D receptor pathway, also known as the VDR. When the VDR is positively expressed, it does two things. One, it regulates gene expression, as noted over here, and two, it promotes responses from intestinal epithelial cells, which regulates the gut microbiome. When the vitamin D receptor is not expressed, as indicated on this side, there is intestinal microbiome dysbiosis, as indicated here. The problem is, when there is a lack of VDR, for some reason that's still unknown, there is a decrease in the anti-cancer response. Now for the importance of vitamin D. Vitamin D, as many people know, allows for us to intake calcium, strengthening our bones, but that's not the main importance in this presentation. First, vitamin D decreases cancer cell proliferation because it promotes apoptosis, which is programmed cell death. It also promotes apoptosis then allows for the regulation of the cell cycle. It also reduces angiogenesis, which is the process of forming new blood cells from old ones. Cancer utilizes this to provide blood flow to tumors so they can have proper nutrients and oxygen needed for growth and metastases. Vitamin D also attacks cancer-associated fibroblasts, which are connective tissues, and lowers their pro-tumorigenic activity. Now onto the gut microbiome, specifically Bacteroides fragilis. There are both benefits and detriments to this. The benefits are only present when this bacterium is present in moderation, when it's in symbiosis with the rest of the gut microbiome. It has immunoregulatory benefits as it increases regulatory T cells and protects against colitis, which is the inflammation of the colon. It also lowers ASD, which is autism spectrum disorder, symptoms in mice. The detriments are present if there are damages to the colon's lining or mucosal barrier, which is usually due to surgery, inflammation, or trauma. When this happens, there's around a 19% morbidity rate. It's very, very common, and it can cause a series of things, including abscesses, which are localized regions of pus in a tissue or organ. It can also cause bacteremia, which is bacteria present in the bloodstream. It also can cause infection and release of bradykinin, which allows for the spread of infection through blood cells. It also releases exotoxins, which could lead to colorectal cancer, and it is known to correlate with very many sinus issues, including ciliary damage, which this is a concern as if there's damage to the cilia, there can be a mucus buildup, and then you'll have a respiratory weakening. There's also inflammation-related hyperplasia, which is the overgrowth of cells in tissue, and there's also metaplasia of the epithelial cells, which is a change in cell type. Now for some immunotherapy terms. First we have CD8+, which as mentioned earlier is a protein on T cells that assists with antigen recognition. Then we have APD1, which is a treatment that currently has an unknown mechanism. Then we have another treatment called ACTLA4, and in order to understand how this one works, we have to first know that CTLA4 helps regulate the immune system, preventing attack. This lowers our anti-cancer response. Now we have things that are found within T and B cells. So these are some genes. The first one that we're looking at is RAG1, which codes for a protein found in T and B cells. It's responsible for the production of T cell receptors and genetic recombination in B cells. Then we have BAT3, which is a transcription factor needed for immune cells. Then IFNAR is a receptor which binds to cytokines, which cytokines are proteins secreted by immune cells in order to facilitate the immune response. Then IF-NGR aids in the immune checkpoint blockade response, and MID88 allows for immune cell signaling, which is needed for pathogen recognition. This article had two main goals. One was to define if globulin strongly acts as a barrier to anti-cancer CD8 plus T cells outside of its acting scavenging abilities with S-GSN. And the second is, how does vitamin D and its relationship with the VDR improve responses to checkpoint blockade immunotherapy? Now for the importance of this research. The importance of this research goes around cancer rates, which includes the prediction that for 2025, where US will have likely 2 million new cases and 6,000 cancer-related deaths, as well with the problems with modern-day cancer treatments, which is mainly that they're expensive, with the average cost being around $150,000 and with side effects affecting the quality of life. Finally, prevention is greatly important as around a quarter of adults in the United States have a vitamin D deficiency. Whether or not this is genetically based, it affects your chances of developing cancer. The article covers many things, as with each question that was answered, a new question arose. The first one was if globulin inactivation affects the anti-cancer response. Then they realized it does, and they wanted to see if it had a relation outside of its acting sensing capabilities. Then it was trying to find if vitamin D displays anti-cancer effect itself, and if this increased vitamin D alters the gut microbiome, and how these alterations boost anti-cancer immunity, and if these alterations are dependent on the functioning of the immune system, and if this improved response can be transferred via microbial transplantation, since they found out that it was microbiome related. Now for the methodology. There are two parts of this methodology. First is in the in vivo portion, and then it's clinical research analysis. For the in vivo portion, there's a few alterations that were done, which is having GC positive and negative, having vitamin D levels deficient, standard, and high, having the vitamin D receptor alterations positive or negative for intestinal epithelial cells, and in order to prevent anything that could have altered by this, they also increased their diet to include more calcium to prevent any things that could have happened by the low vitamin D. Then they also had controls, which were wild type, as well as isotope match controls, the immunotherapies and the immunocomponents mentioned previously. There was some microbial transmission done between the mice, which was fecal transplantation and oral through PBS, as well as co-housing, and in addition of an outside microbe, which includes a Bifragillis and Prevotella brevis through oral gavage. And the cancers observed here were mainly 5555 BRAV, V600E, which is a type of melanoma, and MCA205, which is fibrosarcoma, which is a cancer that originates on fibrous connective tissue. There was also MC38, which is a type of colorectal cancer. Now for the testing. They tested the tumor volume after the trial period ended using the volume formula. They tested the number of immune cells via gating, which is identifying cell populations through flow cytometry. They measured the survival rates, and they did meta-analysis of metagenomic data looking at the genes and taxa via Illumina sequencing, which, as we know, is greatly useful in analyzing microbial diversity through its high throughput capabilities, which we learned at the beginning of this course. Now for the clinical research analysis. There were two studies. This first one had around 2,500 participants, and it tested the vitamin D receptor if it was high or low using chromatin immunoprecipitation. Immunoprecipitation is the use of an antigen that binds to a specific protein to be able to have it form a precipitate during centrifugation, allowing for isolation. Now, there were a couple of cancers that were looked in this study. Skin cancer, which we should note that it has a confounding variable as sunlight both increases vitamin D and also increases the chances of melanoma. Also sarcoma, which sarcomas are cancers that originate on connective tissues. Then there was liver cancer, breast cancer, and prostate adenocarcinoma. And adenocarcinomas are cancers that originate in glandular cells, which are cells that produce and secrete substances. Then this study also had a couple of different testing, including the survival and the tumor stage was taken into account, as well as the response to CPI therapy, which CPI therapy is checkpoint inhibitor therapy. They measured exceptional response as survival past 2 years and rapid progressing as less than 3 months survival. They also looked at the adjusted hazard ratio, which personally I did not understand how that was taken into account at the very beginning, so I looked into it, and it's the ratio between variable and normal survivors, which in this case variable is outside of sufficient vitamin D levels and normal is sufficient vitamin D levels. Now the time between vitamin D measurement and cancer analysis was accounted for by the Cox regression model, and confounding variables were analyzed with the Charleston's commodity index. Now for the second study. The second study had patient data that was like the largest patient data collection for this type of research, and it had around 1.5 million patients involved. They tested vitamin D levels, marking them as deficient, insufficient, or sufficient, but it should be noted that this was only one-time testing, so it is subject to deviations. They looked at a variety of cancers here as well, and they also used the adjusted hazard ratio testing. Now for the results. This shows the base comparison between GC active and GC inactive over time with tumor development. As you can see here, GC active increases tumor volume, while GC inactive has a better anti-cancer response. These graphs show GC inactive's dependency on the immune system. This first graph might be a little hard to read, but if I break it down for you, I think it should be understood here. These two bottom lines are both the controls we were looking at earlier. And then both of these lines, as you can see here, are anti-CD8. Remember, CD8 is needed for the immune response. So it means that without these, our body won't be able to attack cancer. So if you can see, they both have around the same amount of tumor volume. But then you can compare both of the red lines, this one, and both of the blue lines. As you can see here, the red line has a much larger of a difference, as GC is dependent on the immune system. Furthermore, all of these graphs here, with this being used as a baseline, show that when different parts of the immune system are removed, there becomes no difference between GC active and GC inactive, meaning that GC inactive loses its capabilities to enhance the immune response. Here, there are two different immunotherapies as well as two different cancers being shown. If you look here, the red line here and the red and blue lines here are used as a control variable. In both of these cases, the GC negative with the immunotherapy allows for the best percent survival and percent tumor-free areas. Now we have to prove that GC inactive's anti-cancer effect is microbial-based. Here we show when GC positive is separately co-housed, it shows the same representation as before. And this is the GC negative and negative co-housed, and then this is a mix of them co-housed. As you can see here, they end up having around the same, meaning it could be microbial related, but we don't know for sure. In order to see that, we look at a PBS inoculation, and we look at it with just PBS, which is a control. We look at it with a wild type fecal transplant, and we look at it with a GC negative fecal transplant. As you see here, the one that had the GC negative fecal transplant showcases the same thing, meaning it is microbial based. And to further clarify this, we looked at it with a microbiotic, meaning that any changes would be removed. So as you can see here, this light pink line right here ends up being the same. This shows that GC inactive's anti-cancer effect is indeed microbial based. With these graphs, we're looking at GC's dependence on vitamin D. This first graph compares standard and deficient levels. As you note here, also, the red and blue lines will be always used as reference. So as you can note here, once you remove vitamin D from the GC negative areas, they have the same rate as everything else, maybe even higher for tumor volume. So they lose their anti-cancer capabilities. Now here, we're looking at fecal transplantations. So in this first one, the wild-type mice are given normal vitamin D, and it's only the fecal transplant that either has standard or deficient. As you note here, when there is deficiencies, the tumor volume returns to normal, meaning that the mice that they got the fecal transplant from has to have been in a normal or high vitamin D environment. Then here is the inverse. So the fecal transplanted mice had the correct amount of vitamin D, but now it's the wild type mice that are receiving it that have different vitamin Ds. Again, the same thing is present, meaning that the vitamin D has to both be present in the fecal transplanted mice and the mice receiving the fecal transplantation. Now, since we know that GC is dependent on vitamin D, we tested both wild type and GC negative under standard and high conditions. Of course, the expected was that GC negative with a high vitamin D would give the best results, which indeed it did. But something even greater to be noted is that GC negative as well as wild type with high vitamin D gave the same exact response. This now shifts our perspective from GC inactivation being the most important to high vitamin D levels being important. Now we looked at vitamin D immunotherapy results across varying cancers, noted here. So in this first, we used ACTLA4, and as you can see here, the best results are with the high, and that spans across all of them. Here, this final graph, in my opinion, is the easiest to read, as the standards for comparison are deemed insignificant, and you only see the wild type with the standard and the wild type with the high fecal transplantation with the immunotherapy. So of course, as you can see here, the one with the higher diet has a higher percent tumor-free area. Now we look at vitamin D's anti-cancer properties and how they're independent from the immune system. As you can see here, this is a baseline, so there have been no changes to the immune system. Here, there's the removal of RAG1, and the next have other properties that we talked about earlier being removed. As you can see, for all of them, there is a significant difference between vitamin D standard and vitamin D high, with the vitamin D high having a lower tumor volume, meaning it's performing much better, showing that vitamin D's properties are independent from the immune system. Here, we're looking at the vitamin D receptor's role in the anti-cancer effect. As is noted here, with these lighter colored lines now representing the vitamin D receptor that has the IEC negative. As you can see here, that completely removes the response, highlighting the importance of it. Now we looked at three different metagenomic. The first metagenomic data is comparing wild type and GC inactive in a vitamin D standard environment. The second is wild type and GC inactive in both vitamin D high and vitamin D standards. And the final is just looking at the wild type in both conditions. As is noted here, the one that performed the best was the meta-analysis of 2, which is comparing the wild type and GC inactive in their varying conditions, which obviously would make the most sense as there's the greatest diversity from them. As you can see here, there is both a significant gene difference and a significant taxa difference. Previous metagenomic data, we noted that B. fragilis had the greatest change and increase with high vitamin D levels, and P. brevis actually reduced. From here, we looked at P. brevis and B. fragilis in a vitamin D standard environment and a vitamin D deficient environment. As you can see here, P. brevis didn't really change the full effect that much, but an inclusion of B. fragilis greatly reduced the tumor volume, indicating that that is what's causing the anti-cancer effect. Now, in the deficient environment, it's shown that B. fragilis no longer exhibits that effect. That is likely due to B. fragilis being greatly reliant on vitamin D. Now for the results of the clinical research analysis, which will be shown on the next slide. First, we will take a look at study 1. As you can see here, this is comparing the vitamin D receptor signs low and high. As you can see here, when it's more active, throughout all of them, there is a higher % survival through the skin, through the sarcoma, and through the liver cancers. Then, when there is a higher sign, which is noted here for both, there is a lower tumor severity as the tumor is at a lesser stage, which is noted by these bars. Finally, a high vitamin D receptor corresponds to a better response to CPI therapy. As you can see here, you can compare it by the middle lines here. And for both cases, the red, which is indicating a high response, it shows exceptional or standard instead of no response or a rapid disease progression. Then this is using the second study, and it's comparing various cancers with varying vitamin D levels. For most of them, as it's noted here, the line splits off like right here. Vitamin D actually decreases the adjusted hazard ratio because this represents vitamin D deficiencies. So when you don't have as much vitamin D, you have a higher hazard. It is still unknown why these cancers don't show that same result. Now for the discussion. We'll be talking about the advances in the scientific community, further research, and further implications. For the advances in the scientific community, we discovered that the activity of vitamin D involves the regulation of the intestinal microbiota impacting the immune system. The vitamin D and the vitamin D receptor corresponds to various oncogenic factors, including the incidence rate, the percentage of survival, and the response to CPI immunotherapy. For further research, various cancers can be included, as many cancers were included generally in the clinical research analysis, but in the rodent analysis, the most research was done on 5555 BRAF. And it should be noted that since this is a type of melanoma, it coincidentally has the highest amount of confounding variables due to the correlation with sunlight. A larger diversity should be included, as the main ethnicity showcased here was the Danish Central Person Registry, which only included Danish people. And while this was a purposeful choice made by the researchers, as they have a very small racial diversity and they wanted to lower their confounding variables, especially since melanin has a long relationship with the development of melanoma, it would still be both very interesting and necessary to see how these concepts would apply beyond this racial scope. Additionally, varied lifestyles and diets would also affect the vitamin D levels, as lifestyles, including various latitudes on Earth, outside activity, get varying levels of sunlight, and various diets would also alter your vitamin D levels. Also including individuals with genetic predispositions to vitamin D, as some people naturally are able to produce and intake less vitamin D. We could also look into the impact of globulin in humans and the specific functions behind how the microbiome affects our immune system, and exactly which vitamin D receptor dependent intestinal epithelial cell derived factors are involved. Possibly, it's noted that it's possibly because it counteracts the immune suppressive WNT signaling. Finally, for future implementations, we could be using vitamin D, globulin inhibition, or B. fragilistis as a potential immunotherapeutic booster for cancer patients. We could also use vitamin D, VDR, or GC to have earlier diagnoses, as well as to identify people with higher cancer incidence rates. We could use gene therapies to potentially alter GC expression for cancer patients unresponsive to immunotherapies, as well as for people with genetic predisposition to vitamin D deficiencies. And we should definitely be promoting the knowledge of vitamin D deficiencies. As mentioned before, 1/4 of US adults have vitamin D deficiencies. Finally, for the main conclusions, we should note that vitamin D is an essential nutrient, especially for those dealing with or prone to cancer. Vitamin D supplements should be aligned with immunotherapies to enhance their positive results. And finally, having a properly functioning gut microbiome is important to the body's regulation, especially when it comes to the maintenance of the immune system and response. Finally, for some questions to help aid the discussion. Question 1 would be, how do you interpret the double-edged sword that is sunlight, which is the main cause of melanoma but an important source of vitamin D? Question 2 would be, how does vitamin D affect other conditions? Does it affect more than just the gut microbiome? Question 3 is, why do bacteria, like B. fragilistis, have an impact in our immune response? 4 is, what are the specifics of how the immune system works in terms of the topics applied in this study? And 5 would finally be, how do the benefits and detrimental effects of B. fragilistis compare? Because I know this article mainly talks about the positive ones, but I personally found a bunch of detrimental effects, so I just wanted to hear what you guys think. Finally, we just have our references on the next slide. Here are the references, including the main paper, the external figures indicated, and the references where all the statistics and background information were found. Thank you.